What's up? What's up? What's up? Hold on. Hey. Okay, let me get myself on the screen. What's up, people? Oh, I didn't even have my yapping commencing shortly thing up. That's good. You guys already know what's up. You know I like a little silent intro. You know, just fan noises. We're going all fan noises today. Let me see what we're looking at here. What's up, Fishhhh, Mick, SVC? How's it going? How's it going? Sheesh. There we go. There we go. Now, I'm doing water changes this stream, and we're gonna see if I can do it without overflowing a single thing. It's going pretty slow, but that's what's going on over here. Mick says slow start's okay. What you guys been up to? Any good videos? Anything fun? What's up, Johnny? How's it going? Anything I missed? What did I miss? Couple fish gang. Yeah, shout out. Yo, you guys know me so well. I think Jonathan, he commented on the last stream we did where we first talked about these gourami, and he was like, they've been described, and then I saw a couple of fish posted in Discord, they've been described. I'm like, man, the gang is on top of things. We're straight to the scene, man. Let's get these papers going. And then the isopod paper we're going through, that just came out the other day, so holy. Yeah, you like my the orange crayfish? Oh my god, Johnny Best, bro, pulling me out of retirement. Oh my goodness. Message received, Johnny. Thank you, Johnny, for the 10. I better see a bunch of W Johnnies in the chat. Holy. Thank you very much to Johnny. Speaking of which, I have some ideas around membership stuff, so if you're a member, stick around, but there's something I want to do. But dude, Johnny, $10 donation. Thank you very much. I haven't even started talking yet, so let's hope that the stream's good. But I got the orange crayfish from Capital Exotic Fish, and I think they're fantastic. I bought 7 of them, and I put 3 in here, which I think are mixed sex, and I put the rest in my 40 breeder. Couple Fish said he thinks the paper was published last night at midnight. Damn, you're on top of it. Now, will this sell any more of them? So we're gonna have to do this. First, we have to see how many they've sold. What spot? I know one of their top salesmen, Steamford Aquatics. They've been posting about it. Oh man, let's get the Black Arrow Tetras going. All the usual suspects are like, buy these mini gourami. I want to see if it's been working. But maybe them being described, it can't hurt. It definitely can't hurt. Okay, so let's get into things. Wait, let me switch what tank's being filled up real quick. Yeah. No overflowing today. Nope. Not I. Let's switch it up. So, you might notice if I go to a couple tanks today, but as I've not been streaming, I've had a lot more time to work on stuff, and I've slowly been making progress with the fish room looking less shit. So you should notice a general improvement. I've been gravel vacuuming, I've been doing more maintenance. You know, I think there should be a general improvement on how things look around here. I don't want people thinking I got some swamp fatherfish tanks, okay? Wallstead method. Grandma, get out of here. All right, we're gravel vacuuming. I need a certain level of bright sand. What's up, Lisa the Wolf? How's it going? Also, when you can actually see the sand, man, things are a lot brighter. Ooh, ooh. Who knows what the fish is in that left corner? I haven't seen them that much since I got them, but this is my most. This is the only fish I've bought in months. All blurred out in the left corner. I wonder who can get it. Oh, that's so cool that I can see him. I've seen this. I saw this fish for the first time yesterday, and I bought it a month ago, and I hadn't seen it. I thought they had died, but they actually been looking good. I just have rock piles in there, and they seem to love them. Mick guessed humulus. No, absolutely not. I don't even know what that is. I don't even know what that is. All right. So first, talking about this mini gourami. Shout out to the replay crew if you made it through the intro. Shout out to everyone that's trickling in and out. I appreciate you guys sticking through me sticking through me, pause. I appreciate you guys sticking with me even when I'm not as consistent as I like to be. But you know that when we show up, we still got to show up. So we'll continue to keep the work going. I'm not doing anything tomorrow, so we'll have another stream tomorrow. Guaranteed. Okay, so I at least got you today. I got you tomorrow. So put in the Discord your preferred times. I mean, I can't guarantee I'll consider them. You know, plan whatever project you got. We'll do a stream tomorrow. Sunday, I can't guarantee Sunday, but I got you tomorrow. It's not pelvicochromis, and it's not Wallaciochromis. No, it's not. So a little bit ago, we were covering some new fish on stream, as we do way back in the day. And 3 weeks ago, we did new arrivals, and at the time they were calling this Microspherominis, and it was a little mini gourami. Let's see if Wetspot has updated their listing on the fish. And Wetspot seemingly was the first people with them available to us that I'm aware of. Let's see if it's still on their sales page. Maybe now. Maybe the description boost. They've taken them off the sales page. Holy. What was it? What was the name that they had given it? Vietnam. I think that's what it was. Vietnam. I think that was the location. No? Alright. Fish. Livestock. Gouramis. Okay. Sort by latest. Oh, look. They got some chenoides in. Ah, here they are. Out of stock. Oh, shit. Someone bought them all. Okay. There was like 60 last time we checked. Now they're gone. But it was this fish. They were for 50 each when they were initially up. Then they were for 40 each. Ultimately. And now out of stock. So this fish was available. It wasn't described yet. Now we have a description. And that is what we're going to go through first on stream. Kind of get ourselves back up to date. It's not Subacelatus. Nope, that was not Subacelatus, Mick. Alright, so let's get to this description. And you can find this description. I'm going to post it in the chat. But I also put the link to it in the description in the chat of the stream. You can find it there. And now I'll put it in the chat. Perfect, perfect. So, now it's in the chat, now it's pinned. Mick still hasn't guessed the fish, so I win. Now, I kind of am curious, if they come back into stock, like, is the price going to be the same? We'll have to see. I wonder if someone just bought the rest of them, or if a YouTube video was made that really got people interested in buying them. I could totally see that being the case. Maybe they just need one more push, one more video going up. But let's get into this. We got 18 pages, not too bad, not too bad at all. And I'm going to begin. So we have the title here from the Raffles Bulletin of Zoology, published July 24th, 2026. I'm watching my water change, remind me. Mick said Nigeria red cribs. Nope, it's not a crib. I don't. You're in the right zone though. You're close. Close but no cigar, Mick. Nope, not Paranochromis. Nope. I've mentioned these before on stream. And they are sold on Wetspot. So I think you guys will soon get it. I think you guys will soon get it. Alright, so we got the paper here. Nanos from Phronimimus Gemma. So they were calling it Microphronimimus before, but now they have it Nanophronimimus officially. Really classed it up there, you know. We can't have no micro Gourami. It's a nano Gourami. I like the rebranding. This works for me. This definitely works for me. It's not a micro, it's a nano. Perfect. But we got the Nanophronimimus Gemma, and the title says a remarkable new genus and species of miniature osphronimid from Vietnam. Now I gotta look up that word. Osphronimid. Okay, that seems like that's the family that Gourami are in. Alright, well, maybe I should have known that. I didn't do my homework. That will be on the next test. Let's get into this introduction here. The labyrinth fishes of the suborder Anabantoideae, by the way, do I sound alright? Comprise around 170 species. What's up, UAO? Distributed in fresh waters in Africa and Asia. Anabantoideae are divided into 3 families. New Gourami or something? Yeah, that mini Gourami that was $50. We're reading the description of it. The description is free though. They're divided into 3 families: the Afro-Asian Anabantidae with 33 species, the monotypic Southeast Asian Helostomatidae, and the Asian Osphronimidae, the most species-rich family, with 137 species. They're characterized by an accessory air-breathing organ, the so-called suprabranchial organ or labyrinth organ. Drosiclidius and maybe Congochromis? Nope, although that's on my list. I gotta turn this water off in like 5 seconds. So you got labyrinth lungs. It consists of a modified epibranchial one with a complex system of membrane bone extensions, which in its most complicated form resembles a labyrinth. The epithelium of this modified epibranchial one, as well as that of the cavity, is heavily invested with blood vessels which aid in taking up the oxygen from the air that is swallowed into the suprabranchial cavity. The labyrinth organ has enabled members of Anabantoideae to conquer habitats with low oxygen levels, especially swamp areas. The distribution of the most species-rich family, Osphronimidae, extends into Asia from the Indus in Pakistan to the west to Sri Lanka in the southwest, to Amur Basin in China in the east, and to the Sunda Islands in the southeast. I'll turn the water off real quick. No floods this time. No floods. Only fire. Only fire. No more floods. Alright. Okay. The included 14 genera are assigned to the 5 subfamilies. The Bilantinae with the single genus Bilantia. The Osphronimidae with the genus Osphronimimus. The Luciocephalinae with the Luciocephalus. Ooh, those are the pike. Yeah, these are the ones we want. These are the one of the dream fish. The peppermint pikehead, Luciocephalus aura, is a mouth brooder. Very cool. Very cool. Alright. Now see, think about that. This fish is $40, and so is this new nano Gourami. I'm just saying, I'm going with this fish. I'm going with this fish for the 40. Wait, Vic said it. Enigmatochromis lucanusi. I cheated. Wait, how did you get it? How did you cheat? And how did you get it, Vic? That is correct. That's what the fish was. I got a pair in there, and they've just been under the rocks most of the time. I just started seeing them, so hopefully that means they're getting comfortable. I've been sitting in front of that tank when I feed it for a while. All right, we got the Lucocephalinae with Luciocephalus, Sphaerichthys, Paraspherichthys, Synops, the Trigogastrinae with Trigogaster, Trichopotis, the Macropodicinae with Macropodis, Pseudocyphrominus, Malpaluda, Trichopus, Beta, and Pero. Oh, Beta's in there, and Parosphrominus. Looks like I couldn't take it any longer. Wow, how dare you. Alright, materials and methods that they used. Measurements were taken from point to point with digital calipers to the nearest 0.1 millimeter. Due to their tiny size, the specimens were difficult to handle and measure. Therefore, only measurements that could be taken reliably were recorded. Nick said Malpaluda is the funniest name. What's got you so tickled about that name? To count vertebrae and dorsal, anal, and caudal fin rays, 15 species of Nanosphrominus gemma and another 24 members of the suborder Anabantoideae, covering all genera except Sandilia, were radiographed. 2 specimens of N. gemma, female 16.1 millimeters and male 16.2 millimeters, were CT scanned with a Zeiss X-radia context with a voxel size of 4.5 or 4.7 for the body and 3.8 or 3.57 for the head, with an exposure of 2 to 3 seconds and 2 to 3 frames, and 1401 to 1801 projections, with no filter. CT scan data was segmented in Dragonfly, then segmented volumes were rendered and imaged in a mirror. Vertebral and dorsal and anal fin ray counts for the type series of N. gemma and additional anabantoids were taken from digital radiographs. Additional counts came from the 2 CT scan non-types of Nanosphrominus. Pectoral fin ray counts on alcohol-preserved specimens are difficult to attain. The recorded 10 to 11 rays may be 1 or 2 rays too low, as the CT scan specimens which allowed for accurate counts had 12 to 13 rays. We use the term miniature for tiny-sized fishes as defined by Weitzman and Vari. Molecular methods. To place Nanosphrominus in a large molecular phylogenetic framework, we sequenced the complete mitochondrial cytochrome B, partial 12S RNA, complete tRNA, Val, and partial 16S RNA genes, as well as the partial nuclear RAG1 gene of one specimen. The newly generated sequences were integrated into a previously published dataset for the molecular phylogenetics of labyrinth fishes. Laboratory procedures, including DNA extraction from tissues preserved in 100% ethanol, I think that's what it is, ETOH, and PCR amplification of Nanosphrominus, followed protocols by Ruber et al. Using PCR primers described. Additionally, we also sequenced DNA barcoding gene cytochrome oxidase 1 for the same individual, following Ruber et al. 2023. PCR products were cleaned, Sanger sequenced using the same primer pairs that were used for the PCR amplification. What's up, Big Steve? Alright, so this is some of the methods. If you guys want to get into it deeper, the link is pinned in the chat. But let's look at the diagnosis. Nanosphrominus is a new genus. It differs from all other genera in the suborder Anabantoideae by its unique count of XX-XX1 plus 2 minus 3 dorsal fin rays. Sure, why not? Rarely in some specimens of Macropodis ocellatus. Alright, I see. The presence of a series of anteroventrally directed processes off the distal part of the caudal hemial arches. Baudelate's ligament inserting into the ex-occipital. This is almost as bad as when we read the Corey paper. Versus on the basio-occipital, first or second vertebrae, and a unique color pattern in sexually active males consisting of dark brown to black mid-lateral stripe with numerous bluish-green iridescent scales and a blood-red stripe above it running from behind the posterior margin of the eye onto the base of the caudal fin. Nanosphrominus differs from all anabantoid genera except Helistoma. Let's look up Helistoma real quick. The kissing gourami? Disgusting. Alright. They differ from all except Helistoma in the absence of the exo-occipital foramen. Let's look up what the hell that is. It's got to be so exo out, right? This is like a head area thing. Okay. Yep. Exoccipital on each side of the foramen magnum at the base of the skull. I see. Alright. I'm getting concepts of what these words mean. Let's continue. So they differ from all in the anabantoid genera except for Helistoma in the absence of the exoccipital foramen, and from all anabantoid genera except Trichogaster, Trichopodus, Cetnops, Paraspherectes, Spherothictes, and Luciocephalus, and having only 5 versus 6 branchiostegal rays. An additional diagnostic, though not exclusive, character is the miniature size of under 19 millimeters SL, only shared with Paraspherectes lineatus. And we've looked at this fish on stream before. It's another tiny little gourami. I think a little bit before these were available, there was some stuff about the fish, and this was one of the other tiny little gourami that were around. So they're saying this is the only other one that small that they've seen, is what makes it unique, I guess. Etymology: the new genus name is derived from the ancient Greek word, I can't read, it's in Greek, meaning dwarf, combined with the name Osphromenus. The type genus of the family Osphromenidae, to which this new genus belongs. The name alludes to the tiny size of this new fish, which makes it one of the smallest anabantoids today, at Nanosphromenus gemma. Got you. Okay. Now here's some photos of it. Steve said my fish made it to Puerto Rico safe and sound. Damn. Couple fish said it's nanos in Greek. Got you. Got you. Thank you for that. So here is figure 1, Nanosphronemus gemma, holotype ZRC 70244, 18.7 millimeters SL in lateral view. We see a gray beige body with a black lateral stripe with a brown one above it. Here's the description. For general appearance, miniature anabantoid with maximum size of 18.7 millimeters, elongate body, circular in cross-section at the head, slightly laterally compressed to oval cross-section behind the head, but more laterally compressed in the posterior third of body. Head large, almost 1/3 of standard length. Mouth small, superior, snout short. Eye very large, more than 1/3 of head length. Dorsal profile rising sharply from the tip of the mouth to the nape with concave depression between mouth and interorbital area. Dorsal profile continuing almost in a straight line, then descending gradually to a caudal peduncle. Ventral profile descending sharply from snout to jaw articulation, then more gradually to pectoral area, more or less horizontal between pelvic fin base and middle of anal fin, from there ascending gradually to the caudal peduncle. Dorsal and anal fins very long, reaching 50 to 60% of standard length. Dorsal fin with 19 to 21 fin spines and 2 to 3 rays. Anal fin with 21 to 24 spines and 4 to 5 rays. Pectoral fin with usually 11, rarely 10 rays, but see methods. Pelvic fin inserted below base of pectoral fin with 1 spine and 5 rays. The first 4 branched. 5 rays, the first 4 branched. Caudal fin rounded with 1 dorsal and 2 ventral procurrent rays and 6 plus 6 principal rays. Body and head except mouth region scaled in figures 4 to 5. What's up, Minamoto? How's it going? Body mostly transforming ctenoid scales with up to 4 series of steny. How do I say this word? How to say? Nah, there's no. I'm not seeing this word anywhere. All right, someone tell me phonetically how to read this. Say this word. Cteni.
Čtení je moje každodenní.
That's how I'm gonna say it for now. Steny. What's up, Jeremy? Staying missing from scales covering head dorsally, laterally, and ventrally, and also missing from scales on dorsum along dorsal fin, ventrally from shoulder girdle along anal fin base. So that scales appear cycloid. 28 to 29 scales in a lateral series, 4 to 5 scales between lateral series and dorsal fin base, and 5 to 6 scales between lateral series and anal fin base. Base of dorsal and anal fin spines covered by a series of small elongate cycloid scales. Basal fourth of caudal fin also covered by small and thin cycloid scales. Lateral line canal restricted to head, no perforated lateral line scales on body, only 3 lateral line pores associated with frontal, 3 with extrascapular, none with pterotic, 2 with lacrimal, and 5 with preopercle. I gotta look that up later. Suprabranchial chamber extending posteriorly to the level of the third vertebra, housing simplified labyrinth organ formed from modified epibranchial 1. All right, I understood all of that. Coloration and preservative. After formalin preservation, body coloration cream with 2 stripes mid-lateral. As we can see here. Yes, I would say that's accurate. Mid-lateral stripe, 1 and 1.5 scales wide, extending from snout through middle of eye, posteriorly to base of lower of caudal fin, forming lower caudal spot. Dorsolateral stripe, lighter brown or reddish brown, half to 1 scale wide, extending from dorsal posterior corner of opercle to upper half of caudal fin, forming fainter upper caudal spot. 2 specimens selected for CT scanning and preserved in ethanol with much darker overall coloration than formalin preserved species. A female identified by eggs and body cavity with more prominent mid-lateral stripe and less conspicuous dorsolateral stripe, otherwise with numerous melanophores above and below mid-lateral stripe from head to caudal fin base. Middle scale rows 2 and 4 above mid-lateral stripe. And here we see the specimens in question. With the melanophores and middle of scale producing 3 narrow parallel stripes. Body below mid-lateral stripe with numerous melanophores but less densely distributed. Melanophores also present along pectoral fin rays. They got a putative male, no eggs, and body cavity with very dark coloration in figure 2. So they're saying we got the female on the top here, and then what they're saying is a male on the bottom. And you can definitely see a big color difference. So maybe these aren't too hard to sex when they're alive, I don't know. It seems like when the males are sexually active, probably it's not too difficult to tell. Let's see, coloration in life. Putative female, similar in color pattern to formalin preserved specimens with prominent mid-lateral stripe, but with series of blue-green iridescent spots in each scale. And iridescence on post-orbital areas, so you want to tell the difference between males and females, that's the part we're at. Dorsolateral stripe not clearly demarcated from brown dorsal third of body, but black mid-lateral stripe and brown dorsal area separated by light cream interstripe. Dorsal and anal fin spines light brown, caudal, pectoral, and pelvic fin translucent. Putative males overall darker with more iridescent scales on mid-lateral stripe and area below it. And we'll see that coming up in figure 3b. Actually, let's just look at figure 3b. So figure 3b, we see the male here. Definitely got some licorice gourami vibes. Just with some short fins. Because the licorice gourami I have much longer pointed dorsal fins, I feel like. Okay. Yes, 3b, which we just looked at. Dorsolateral stripe, red. Dorsal and anal fins, dark, with iridescent margins. And before, remember they say that the pectoral, pelvic, anal fin spines, and caudal are translucent in the female. So over here they're saying dorsolateral stripe, dorsal and anal fins, dark, with iridescent margins, sexually active males, with very dark, almost black lower 2/3 of body, and head covered in numerous blue-green iridescent spots from sub and post-orbital area to caudal pedunctal. Dorsolateral stripe 1.5 to 2 scales wide. A bright neon red from behind eye to caudal peduncle. Dorsal most area of head and body black, separated from dorsolateral neon red stripe by a narrow lighter stripe. Base of dorsal and anal fins also with numerous specks of blue iridescence, both with fins with iridescent margins, pectoral fins haline except with small melanophores lining margin of fin rays, pelvic fins dusky to black. Mick, yeah, I think that's true. I think I saw the same. Alright. Etymology, the species name is derived from the Latin word for jewel, gemma, alluding to the stunning coloration of sexually mature males. I think we'll see people breeding these relatively quickly. Remark, our aquarium observations show that during reproductions, male Nanospherinus build a small bubble nest underneath overhanging structures such as plant leaves in which they embed their eggs. Nests are guarded by the male. So pretty typical stuff. And we see live specimen photos of a male. Definitely feel like you can tell the difference between the male and the females pretty readily. I'll definitely say that. Alright, what else? We got anything else in here? We got some pictures. This is a rendered CT image of the suprabranchial chamber. That's pretty freaky. Epibranchial one in view. Damn. Look at that. Look at that. And we have some more photos of them breeding. A mature female with eggs in figure 1. Well, in figure A. That's what a mature female with eggs looks like. Then we got a courtship position. We got bubble nests under leaves. And that male does look pretty sick. Now, the question still is this a $50 fish? Well, if it's easy to breed, you might get a wave of being able to sell it for a pretty good price coming up. So, and who knows when they come back in. So if they don't come back in for a long time, you could probably make a good amount of money breeding them. We'll have to see. Have to see. But all right. Ooh, look at this. We got a little family tree of. What's your? Oh, what is this? Belonatia? What is that? That looks like a cichlid almost. Belonatia. Referred to as a comb-tailed gouramis, the only genus within the subfamily Belonatinae. Huh. I mean, this photo, it doesn't look that beautiful or anything, but it's kind of a cool fish. It looks like a gourami mixed with a cichlid almost. Huh. Yep. We got the stenops, the luciophallus, yep. Spherictes, Trichopodus, Macropodus, Malpoluta. Those look kind of crazy. Malpoluta. It's a paradise fish. Malpoluta cristeri. Ooh. That's kind of cool. It's like a streamlined paradise fish. Very betta-esque. Very betta-esque for sure. Man, they just keep making fish for us to want to buy. The fins are just exquisite. Even without the colors, just very long, elongated. Alright. I'm sold. Mick said a friend bred Malputa 10 years ago over here. Damn. Alright. Add this fish to the list. Wish list. Big wish list. Betta, Paris Ferminus. And now we have the new one, Nanospherinus. Dorsoclidius said I want those immediately. I mean, this is the best part of the paper almost. This like a little diagram to give us more lust for fish. Oh, they said this is Belantia signata. And then what's the Malputa that they used? Because, okay, that was the one that we looked up. This is the one that they reference. I'd pay 100 for a pair, but not 300 for a group. I'm with you, Big Steve. Man, we need the Malputa. We need the Malputa. And you said that name was funny because in the Swedish kids TV show they used the word peluda, meaning little mini me. It almost sounds like the Swedish word for fart. Oh, fantastic. Big said I want it all. Nope, no buying fish for me. Alright. I've been good. We're sorting out stuff. We're focusing. We're getting grow out space. My Epistogramma Nisanai juveniles I'd been raising up. I had 4 of them, 3 females, 1 male. I was saving them to bap them. But Chad needed a male, so I gave him a male. Turns out the male left behind some fries, and I gotta get the other females out of there. I did not know. Well, I kind of could tell that they were getting to the size where they'd have babies, but I didn't know it was that imminent. Makes it look at the schedule. The numbers refer to how closely the species are to each other. Oh, I see. You're saying these numbers here? Okay, well, like, what does a bigger number mean they're closer together? I have to look into what that means more. But I thought this was a great description paper. A lot of them don't always have such great photos of the fish alive. Like, I don't think the Black Arrow Tetra paper, I don't think it had photos of the fish alive like this. I mean, this is pretty fantastic. I'm not gonna lie. Highly recommend this one. Alright, let me change the camera real quick. Where do I want it? Actually, I don't know. I'm kind of liking the Histatus. I'm liking the Histatus vibes. Never mind. Never mind. I reversed myself immediately. Okay, let's get into the isopod paper. Let's get into the isopod paper. And by the way, shout out to the Discord. If you're not in the Discord has been increasingly alive recently. In big part thanks to the live bearer people. I've not been that much into live bearers, but man, how much the live bearer people are into live bearers does make you want to get into them. So I feel like I'm warming up a little bit, but I need to have a bit more success with the ones I have. That's the problem. If I could get these like green, Moscow, whatever they are, my females are big all the time, but I just don't get a lot of fry from them. If I could get those going, then I might actually join you. But I've just been frustrated because the females are huge. I think I've gotten like 2 fry that have come out of them. I don't know how it's possible. People are like, I don't know how to stop it. I just have a billion guppies all the time. All my guppies they just don't drop that much fry. And I've been feeding them good stuff too, and they get huge. So I don't know what's going on. Anyways, that's something it's on my to-do list of troubleshooting. But shout out to the Discord. I put the link to the Discord in the chat. You guys should be in the Discord and posting photos and stuff. I've been trying to post more photos and breeding project stuff in there. So if you want to see what everyone in the chat is up to and working on, that's the place to be, man. And it's definitely one of the best places for aquatic isopod nerds. We had Christian's up in there posting photos from isopods he's been collecting. Very cool stuff. So, we gotta do a few more isopod papers more recently. Mix it, yep, library people are awesome on the genetics and logic. Yeah, I need to get better on genetics, and then I'd be able to handle it a bit more. Alright. Reject optional cookies. So this was published the 22nd of July. So we're only doing things hot off the presses here. I'll put the link to this in the chat. And it's another genetic paper, so shout out to SVC I'm hoping SVC absorbs a lot of genetic stuff and understands it so he can explain it to me. I feel like eventually he'll pop out with like a graph of a billion different fish or shrimp or whatever. He's gonna like just like he did with the quail. He's gonna have it all figured out. But the title of this paper is Genetic Commonalities in Eye and Pigment Evolution in the Asellus aquaticus species complex extend to biogeographically and ecologically most divergent cave population A. infernus. A. infernus. Wow, the first thing that comes up is a car from GTA. Now that's just not right. Minamano said hybrid cichlid leaders as well, since they work with multiple species and genera. Hybrid cichlids? Nah, that's evil. Hybrids are evil. No. The Asellus aquaticus species complex, a freshwater isopod crustacean, includes surface and cave ecomorphs that differ markedly in eye and pigmentation phenotypes. Previous genetic studies in Slovenian cave populations revealed that some genomic regions underlie eye and pigment loss across multiple cave populations, suggesting strong genetic constraints on these traits. To test the generality of this pattern, we examined Asellus infernus, a cave from Mangalia, Romania that inhabits in thermal sulfitic waters and is geographically distant from the Slovenian cave populations. Based on its distinct ecology and likely different ancestral surface population, we hypothesized that A. infernus would rely on different genomic regions for similar traits. Using hybrid crosses, there you go, hybrid crosses, we uncovered novel hybrid phenotypes unique to A. infernus, highlighting the presence of additional genetic variation not observed in the Slovenian cave populations. However, we found that the same genomic regions previously implicated in Slovenian populations were also associated with similar eye and pigmentation phenotypes in A. infernus. Eye and pigment phenotypes examined were pigmented versus unpigmented individuals, orange versus red/brown, and red versus orange/brown eye pigment, stellate versus diffuse head pigmentation pattern, and presence versus absence of ommatidia and/or their fragments. These results point to shared genetic mechanisms of eye and pigment reduction between different cave populations. All right, let's get into the full thing. And that was just the teaser. Holy. Mick said, I appreciate a good mathematic explanation for my cichlids, even though I rarely prefer man-made hybrids. I hear you, Mick. I hear you. All right. Introduction. Here we go. The repeated evolution of similar traits has been documented across many biological systems, such as stickleback fish and old-field mice. Historically, uncovering the genetic mechanisms underlying repeated evolution has been challenging, as many species displaying these patterns are non-model organisms or emerging models with limited genomic resources. However, advances in sequencing technologies and molecular methods now allow genetic investigations beyond the traditional systems. Comparative studies reveal different possible outcomes. In some cases, the same genetic mechanisms are repeatedly recruited, such as the involvement of MC1R in pigmentation differences. In some cases, different mechanisms give rise to similar traits, such as in Bahamian mosquito fish. Distinct mutations in different genes may be more likely when genes are not constrained by pleiotropy. I'll look up that word. Pleiotropy, a genetic phenomenon where a single gene influences multiple, seeming unrelated phenotypic traits. Interesting. What's up, Captain? How's it going? While different mutations, we just got started on the isopod paper share in time. While different mutations with the same gene may occur at mutational hotspots, conversely, identical mutations in the same gene across populations may indicate that standing genetic variation in the ancestral population drives repeated evolution. Cave animals provide excellent systems to study repeated evolution, as many species have independently evolved convergent traits such as eye reduction and pigment loss. Because cave adaptation occurs in multiple independently evolved lineages across the tree of life, one can examine repeated evolution across different scales, between distantly related species, e.g. shrimp and fish, between closely related species, e.g. two terrestrial isopods, or between populations within a single species or a species complex. The best studied cave animal in this context is the fish Mexican tetra, Astyonyx mexicanus. Work across multiple populations has shown that mutations in the same gene underlie albinism in independently evolved populations, while additional pigmentation phenotypes are controlled by other loci. Eye loss may involve both similar and different mechanisms across populations. An emerging system for investigating repeated evolution of cave-associated traits is the Asellus aquaticus species complex, a freshwater isopod with both cave and surface ecomorphs. Come on, model specimen, let's go. Distributed across Europe. I don't want to hear anyone talking about Asellus aquaticus in America. I'm going to slap you. Okay, through this paper, we follow the population designations established by Protas et al. When referring to distinct cave populations. Previous studies in Slovenian populations have identified large effect genomic regions associated with eye and pigment loss. These populations span from a range of geographic and phylogenetic distances from adjacent parts within the same cave system to more distant and distinct cave systems. Remarkably, across all populations examined to date, the same genomic regions have been implicated in eye and pigment traits. This striking genetic parallelism is evident even when the trait itself is not expressed in cave animals. For example, orange pigmentation segregates and crosses of distinct cave populations with surface animals, despite not being visible in the cave isopods themselves because it's masked by a loss of pigmentation. The cause of this remarkable genetic constraint remain unresolved. Several factors could influence whether the same or different mechanisms underlie repeated evolution. Cave populations derived from the same founding surface population shared its standing genetic variation and might have recruited the same alleles during adaptation, whereas those derived from different founding lineages and gene pools may be more likely to fix alternative alleles of the same or different genes. Similarly, environmental context may shape evolutionary outcomes, although all subterranean habitats, including caves, are characterized by the absence of light. It is increasingly recognized that they may differ in other environmental factors such as temperature, humidity, food quality and quantity, and overall environmental stability. Populations adapting to ecologically similar caves may rely on the same genetic mechanisms, while populations in ecologically divergent caves might require different genetic solutions, especially if pleiotropic or linked alleles must simultaneously respond to multiple selective pressures. To date, all studied cave populations in the Slovenia of the A. aquaticus species complex are located within 100 kilometers of each other. However, the Slovenian caves examined so far share broadly similar environments underground, stretches of sinking rivers with non-sulfidic waters, seasonal temperature fluctuations between 7°C and 13°C, and comparable food resources. However, here we extend this framework to Asellus infernus, arguably the most divergent member of the aquaticus species complex. A. infernus inhabits a groundwater system near Mangalia in Romania, over 1,000 kilometers away from the Slovenian populations. This geographic separation is mirrored in phylogeny, whereas the Slovenian populations belong to the northern Dinaric clade. A. infernus is part of the Central European clade, and thus clearly derived from a different founding surface lineage. Beyond its phylogenetic distinctness, A. infernus is set apart by its striking ecological conditions. Its subterranean habitat is characterized by highly constant sulfidic and thermal waters. These extremes also shape its biology. Individuals feed primarily on sulfur-oxidizing microbial biofilms and face comparatively limited predation, from the e.g. from the water scorpion Nepa anophthalma and amphipods, Nebahargus rakovici and Nebahargus dankoi. The fudge is this? Water scorpion? No, not my isopods. Oh no. Yeah, that looks nasty. What's one of these amphipods? I'll be eating them. Got a photo of these. Ah, it looks like a scud. I knew it. God damn it. Proven right again. All right. In contrast, Slovenian populations consume detritus and biofilms and experience stronger predation pressure from the Proteus anguinus and multiple Niphargus species, and possibly leeches. Although A. infernus has historically been challenging to study, recent work has established transcriptomic resources, enabled comparative embryology, and produced F1 crosses. Complementation crosses further suggest that the same genomic region underlies both pigmentation loss and orange pigmentation in A. infernus and Slovenian populations. However, additional genotype-phenotype associations have so far not been possible without generating F2 hybrids. In this study, we test whether the remarkable genetic parallelism observed in Slovenia cave populations of A. aquaticus species complex extends to A. infernus, a biogeographically and ecologically distinct cave population. Specifically, we generate F2 individuals from A. infernus crossed to the surface ecomorphs, describe their eye and pigment phenotypes, and assess whether they are associated with the same genomic regions as Slovenian cave populations. I wonder if any of this stuff will be interesting or relevant to some of the albino isopods that have been popping up for Captain every now and then. I haven't really had any. I've had some that are more yellow and shiny than others, but I haven't really had. Maybe we need to do a isopod trade. I need some fish I can send you one day. Okay. Specifically, we generate F2 individuals from A. infernus crossed to surface ecomorph. Yep, I read that. Materials and methods, animal sampling and husbandry. Catgirl said, "Whoa, nerd stuff." Come on. You know what this channel is. For this study, we collected a cave and surface ecomorph of Asellus aquaticus species complex from the vicinity of Mangalia, Romania. Around 30 individuals of each were collected. The cave ecomorph, Asellus infernus, was sampled in May 2021 from Avram Enku and Dimitru Anna, both characterized by sulfidic thermal water, 21 to 23 degrees Celsius. The surface ecomorph A. aquaticus was collected from the Turkish Bath Stream in July 2018 and Limano Bridge Stream in May 2021. These latter sites are located along the shore of the same pond and were treated as a single panmitic population. Let's look at panmitic. Population biology where all individuals can freely interbreed, and any male-female pair has the equal chance of mating. Panmixia. Uniform random fertilization, which means individuals do not select a mate based on physical traits. Alright, interesting words. Captain said the albinos are breeding and producing more albinos is probably a het recessive mutation. Oh, damn. When you get an albino line, I'm going to have to purchase. Animals were maintained following the protocol of Protas et al. 2011. Fed algae pellets and reared in artificial Daphnia water. Both ecomorphs were kept under identical laboratory conditions. We first established a classic cave X surface F2 cross. Cave males of A. infernus were crossed with surface females. Wow, surface females? Uptown pod. Uptown pod. You know I'm in love with an uptown pod. Artificial Daphnia water. I don't know what artificial Daphnia water is. They were reared in artificial Daphnia water. Yeah, why did I not question when I read that more? What the hell? Captain, you need to get this artificial Daphnia water in stock, okay? So they crossed the cave males with surface females. In total, 3 different surface females and 3 different cave males were used. Each cross had a unique set of parents. 3 initial F1 crosses were produced, which in turn yielded 8, 9, and 10 F2 families, respectively. In 2 of these initial F1 crosses, the surface female parent was likely heterozygous for the orange eye pigmentation phenotype. The surface female parent was likely heterozygous for the orange eye pigmentation phenotype. These F1 animals were originally generated to test whether the orange pigmentation observed in different populations was controlled by the same or distinct genomic regions. In total, 120 F2 individuals were obtained ranging from 4 to 8 months of age when harvested for phenotyping and genotyping. Phenotypic variation was pronounced in the classical cave X surface F2 cross, making certain traits such as orange versus red eye pigmentation difficult to distinguish. Generally, red and orange are distinct phenotypes, but in the background of additional pigmentation differences, some individuals seemed intermediate between the two phenotypes. Catgirl said, "Hey guys, hope everyone's Friday was good." Hope your Friday was good. One possible reason for this is that multiple eye and pigmentation traits could exhibit epistatic interactions, unequal interactions between different genetic loci. Notably, pigmentation phenotypes were more diverse than those previously observed in Slovenian populations. To reduce potential confounding from epistasis and clarify phenotype-genotype associations, we generated additional crosses using F2 individuals of specific phenotypes back crossed to surface individuals. So here we have a graph on the screen. So over in figure A, we see they had an A infernus male with a surface female. They got the F1 hybrids. Those got together, F2 hybrids. Now, you see they're representing the cave male as having no pigments. So it looks like they said the F1 hybrids all had pigment. Then the F2 hybrids, we see some orange. Man, we need to cross some. We need to do some experiments with it. They said one's red, and we got red. Well, that might just represent the eye pigmentation, actually. So we got one red, one orange, 8 brown, and 2 that have the cave, no pigment over here. Then they took a red with no eye female, so no eyes coming from the cave male probably. What's up, DP Exotics? They took the F2 red, no eye female, crossed that back with a surface male. We had the F1 hybrids. Then we had the F2 hybrids, would look almost all just brown, but they had 2 of the reds in there. Captain Critter's a caveman. Yes. So the F2 individuals from the surface female and cave male to the caveman to surface female hybrids, and then intercross to generate F2 hybrids, those exhibited high variation eye and head pigmentation. Yeah, we see a lot more variation in this cross. This one only had like 2 that had the red. So the F2 of those exhibited high variation eye and head pigmentation and eye phenotypes. And B, the red eyeless X surface female X surface F2 cross. An F2 female from the classical cross. The red eye pigment, no amotidia and or amotidial fragments were crossed with the surface male. The resulting F1 hybrids were intercrossed to produce F2 offspring. Phenotypes back-crossed to surface individuals. Among these, the red X eyeless surface F2 cross. 13 families were established, which subsequently yielded 57 F2 individuals. These offspring were harvested at 4 to 12 months of age for phenotyping and genotyping. This is a long-term thing. Fathom Aquatic says, I was promised mini Gouramis. Yeah, we already did that. That's what you get for being late. This is isopod time now. Go cry to your Bashur, or your Bicher, or your Biker. Anyone call them bikers? Anyways, as I was saying. So, phenotyping. Animals were first anesthetized. They were slaughtered. They killed the isopods in a clove oil solution. 50 milliliters of artificial Daphnia water with 20 microliters of clove oil. Oh my god, bro. They killed them with the clove oil artificial Daphnia water combo. Then they rinsed them twice in artificial Daphnia water. What is this stuff? Each animal was then photographed under a Leica S8 APO microscope for phenotyping. DP Exotic said always killing them. Hey, it's the way you gotta phenotype them, I guess. Hey, do you support it? I mean, look, I don't support it, but Fathom said they got the Dan's fish treatment. Hilarious. Maybe Dan's on to something after all. Heads were scored for the following qualitative traits. Can you imagine someone kills you and scores your head for science? Holy. 1. Eye pigment, brown, orange, red, no pigment. 2. Head pigment, present or absent. 3. Head pigmentation pattern, stellate or diffuse. 4. Eye structure. Number of ommatidia or ommatidal fragments on both sides of the head. Cat Girl said, where's the cruelty-free data? Yeah, I don't... That's not... Yeah, Minamoto said that doesn't exist, yeah. There's no cruelty-free data. DP Exotic says, I don't know what fake Daphnia water is. I don't... They keep referencing artificial Daphnia water. Like, I need an explanation. Using the phenotypic data, we later tested the following genotype-phenotype associations. 1. Presence versus absence of pigment. Present indicated any pigment in eyes or head, while absent indicated no pigment in either. 2. Orange versus red/brown eye pigment. 3. Red versus orange/brown eye pigment. 4. Stellate versus diffuse head pigmentation pattern. 5. Presence versus absence of the ommatidia and or ommatidal fragments. Individuals were classified as eyeless if they had no ommatidia or less than 2 ommatidal fragments. Often unilateral, occasionally with a small bump counted as a fragment. Okay. Ambiguous phenotypes were not included in the phenotype/genotype associations. After phenotyping, heads and telsons were preserved in 100% ethanol, while the rest of the body was retained for DNA extraction. Daphnia water is what Nick drinks after stream. Well, actually, I'm going to start drinking my kombucha. If you have something, if you're drinking a water or whatnot, Friday night, thank God it's Friday. You can clink the screen in 3 seconds with me. Cheers. 3, 2, 1, clink. Clink. So, we continue. Whew. The results. The classical cave X-surface F2 cross generated by mating cave males of Asellus and Furnus with surface females of aquatius produced F2 hybrids with highly variable pigmentation. Yep. When pigmentation was present, it differed in color in the eye, brown, orange, or red. And the pattern. All right, we read some of this kind of stuff. It's kind of repeating. Let's see. We kind of read the results a little bit. Catgirl said, I clinked my berry hibiscus tea. Very nice. Mick said, BRB needs some coffee. All right. Clink, clink, clink. In this study, we crossed the individuals from the Romanian cave population A and Furnus with surface A aquaticus to generate F1 and subsequently F2 hybrids. Genotype-phenotype association tests revealed that the same genomic regions previously linked to the eye and pigment phenotypes in Slovenian and cave populations are also associated with the same traits in A and Furnus. This finding is striking given the considerable biogeographic and ecological distinctness. For the phenotypes of pigment absence and orange eye pigment, the results were consistent with those from the Slovenian cave populations. The pigmented versus unpigmented phenotype and genomic region marked by scarlet shows significantly and similarly strong association in A. Furnus. Complementation crosses provide further support. A cross between subterranean rack hybrid and A. Furnus, as well as crosses between multiple Slovenian cave populations, indicate that the same gene likely underlies pigment loss across all populations examined. DB Exotic said, I put a screenshot in the Discord of how to make the fake Daphnia water. It looks like it's water, water, and water. Oh wait, trace sodium selenite solution, potassium chloride, sodium bicarbonate, magnesium sulfate, calcium chloride. Holy. We need this fake Daphnia water. Fab says, whenever I need to humanely euthanize aquatic life instead of clove oil, I prefer adding them to a tank where I apply the father fish method. Oh, I gotta do some investigation later. Mexicalia wanted me to look at a channel. I should do that. Okay. Females used for F2 crosses were heterozygous for orange allele. Previous complementation tests further support that the same gene likely underlies the orange pigment across. Alright. Oh, there we go. This is the photos. I was trying to get to the photos. There we go. So, you want to look at the eyes phenotypes? Now we're cooking. This is where there's something I can comprehend here. I mean, I've been understanding everything this whole time, actually. Sorry, I misspoke. It's just I like photos. That's all. That's all. So, figure A, we have a surface individual showing 4 amatidia. Let's just look that word up real quick. Small independent optical units that make up the compound eyes of arthropods like insects and crustaceans. The compound eyes of arthropods like insects and crustaceans are comprised of units called amatidia. Okay, that's basically what I figured it was, but I didn't know how to say it. So this is what 4 look like in figure A, if you can see that. What the hell? And a brown pigmented eye spot. No, wait, sorry. That A is showing none. So B, this is 4 with a brown pigmented eye spot. C to E is a mixture of normal and fragmented amatidia. F to J is a complete absence. Okay. K to O, complete absence, presence of only 1 to 2. And C to E is fragmented amatidia or a mixture of normal and fragmented amatidia. Man. So different amounts of I happening here. Minamano said, I want to get aquatic isopods again. I failed twice. Oh, damn. You got to post the setups and troubleshoot with people. Ooh, we got another graph. Genotype, phenotype associations in F2 individuals of classical cave X surface F2 crosses. So for those that know how to read this, you can read this. We got all sorts of numbers here. F2 hybrids were either homozygous for the cave allele, CC, heterozygous, CS, or homozygous for the surface allele. Kramer's V estimates the strength of associations between genotype and phenotypes measured. Alright. What else we got in here? Some more of the eyes. And some crazy photos, not gonna lie. Ooh, now we can actually see the face a bit. And these more zoomed out ones. The PDF is like lagging like crazy right now. But check this out. Look at the little eyes. Oh, and you can see the red eye pigment. Versus, I see what they're saying, versus orange. It's so freaky. The little isopod face. Oh my goodness. It's kind of wild. So some of them crossed with the eyeless ones. Kind of had a little eye, red eye, all sorts of different eyes. Damn. Let's see. Because our current genotyping cannot distinguish between homozygous and homozygous states, such states could produce misleading results. Resolving this will require methods capable of discriminating among all possible alleles states, as well as sexing the F1 hybrids. Alright. Overall, the genomic regions previously identified in selenium populations appear to also play a role in Infernus. However, it is likely that the additional regions might also be involved in these traits. Alright, alright. Guys, this paper is linked in the chat. I definitely recommend you look at it for yourself. I'm pulling up this chart because this is the most readable part of it for me. The photos. In the flesh aquatics. What's up in my tanks and everyone in the chat? What's up in the flesh? How's it going? How is it going? So you're telling me I have to look at them in the eyes to see if they're true to me? Sometimes that's the only thing you can do. Now, I've done that. We've done the gourami. Now, let me look at what Mexicali sent me to look at, alright? You guys need to help me with this, because my brain power. Tomorrow, I'm going to go to sleep early today, so I can come back tomorrow strong. Alright, I'm going to come back tomorrow so strong. Watch me not come back tomorrow at all. But he sent me an email. Alright. Sent me an email, and he wants me to look at a channel to see if it's legit. Alright? I think that if a couple of fish is around, he'll be able to tell me if it's legit. Said an interesting channel, Hillstream Kingdom LLC. After watching a few videos, I found a few interesting facts. Apparently, it's a business trying to import cool fish from Asia. One of the co-workers is a guy from Maine. Funny thing is, he calls himself Dr. Sam Fernald. Oh, is he a real doctor? We'll have to find out. Check out their website, see if it seems legit to me. It seems sus. All right, we're gonna take a look at this. Maybe it's just a new business with a terrible website. Have you guys seen this? A couple of fish said their ID isn't good, but they're pretty popular for hillies. Nice. Let's take a look at what they got then. Oh my gosh. We're out in the wild with it.
Trust me, I never thought I can find so many fish here.
It's absolutely the paradise of nature.
This stream is unbelievable. Road, tea field, even tourist temple. Human activity.
Alright. I just want to see the fish.
Colored iridophores, which are scales which trap and reshape ambient light into color and hue.
At this point, I'm going to hand things
All
over
right.
to Dr. Sam.
He said it's legit.
We're already seeing the wild side of Sodogastomys and Laticeps.
We got t-shirts.
Now, let's see the same
Man,
fish
these hill
from
streams
a
look freaky.
different
Okay,
perspective.
what's going on with this?
Homochorium.
There we go. Okay,
Sam
now
has
we're
been
seeing the
keeping
fish.
and
Thank you.
breeding this species for years.
Oh, yeah.
And honestly know them
Oh,
a
yeah.
lot better than I do.
Holy. Catgirl said the suspenseful music is kind of funny to me. DPXI says I'm planning on buying hill streams from them. So would you buy hill streams from them, or are you saying the ID isn't reliable?
Let's talk about pseudogastromites.
Also, did I see you say you think that the Swellia are a different Swellia than
So
Asellus?
historically, there were two subspecies of this guy, Pseudogastromyzon liangjiangensis.
Mikim said early, so you're saying this is only a 5-hour stream?
True laticeps, which are
Bro, I'm not doing a
fish that
5-hour
have
stream.
Purple.
I need to do some
ranging from
exercise. That's what I want to do
blue all the way
tonight
up
before I
to
go to
like a
sleep.
royal purple and sometimes purple red, depending on the location
All
where
right.
you catch. Again, I think that
Let's look at the website. Hillstream Kingdom. Hillstream loaches, gem scales, and more. Rare and imported from China and beyond. Alright. Aquarium pets. Vertebrates. Now, I just need a Hillstream button. Where's the Hillstream button? A couple fish said, I have not, but I'm listening to Chinese stuff. Sam likes to make up trade names low-key. Alright. Cacro said 4-hour stream. No, I'm about to get off any moment. Everything's out of stock. Red Spot Stout Hillstream Loach. Yeah, what the terrible name. Okay. You said that they're pretty popular, but where's the name? Ah, here we go. What are these names? Aquafinned Rainbow Hillstream Loach. How many adjectives can we fit? How many? Oh, and we still got the WordPress favicon. Come on, can we please I just put the barest of minimum effort into the websites. You see at the top here, it still got the WordPress favicon on there, man. They ain't customized the damn thing. Please. Please. Yeah, this is crazy. Oh, and they're breeders. Oh, they're breeders too. Only $25. It's kind of affordable. Breeders. Alright. Buys King Hillstream Loach. Jade Belted Casino Hillstream. What the actual hell? See, I could never buy anything from this person based off these names. They just offend me on a moral level, on a spiritual. What in the hell is this? Jade Belted Casino Hillstream. I mean, is this like a random name generator at this point? I can't believe a human put this together. If they did, what's wrong with you? Let's go to the casino. Now, see, this is the most normal one. And it's kind of lazy, but it works. It works. Lime Borneo Sucker. Gastromyzon SP Lime. It's green. I get it. I get it. Jade Belted Casino Hill Street. I'm not getting it. I'm really not getting it. Okay, we just got a bunch of names. I mean, if they actually have these fish, they look pretty cool. I see why you order from them. But it is kind of like the site is making me feel like I can't trust anything that's on it. I can't trust that I'm gonna get what's in the photo, and I can't trust that I'll know what the hell I mean, when I see show quality breeders on hill streams, I mean, maybe they do hill stream shows elsewhere, but I don't know what they're even talking about. $250 for Pseudogastromyzon fasciatus O River breeders. So does this mean that it's a pair? Is that what it means? I mean, I guess ordering a pair is kind of nice. Pink Pearl Fin Hillstream Loach. Now, see, this is what you... let me help you. Green. Pink. Watermelon Loach. Watermelon Hillstream Loach. You're welcome. Okay. What else? Strawberry Lime Loach. Strawberry Lime Hillstream Loach. I mean, come on. Let's just do something. Let's just do something accessible. I don't know if I'm going to call this the Pink Pearl. Alright. It looks cool. I see why Mexicali found the website suspicious. Captain Critter said, culling albino pods that I listen in. Interesting, most of the culls have pale legs. Sharing pics in the Discord. Okay, I'll have to take a look. These fish are harvested in the thousands in China. No one's trying to protect anything. He's translated the Chinese characters, which are notoriously long-worded. Ah, I see. Well, there you go. It's not his fault. Diana said, whoa, that's beautiful. Ooh, spring loach. I like that. So, couple fish. You haven't bought from them. One, why? And two, like, can I trust if I buy from them, I'm going to get whatever's pictured? You said that they're popular, so they have to have a good reputation. DP Exotic says this website hasn't even been up for 5 days. At the bottom, 2026 Hillstream Kingdom, all rights reserved. Oh, someone put their name on this design? Holy. Okay, I mean, everyone's got to start somewhere. Website created by Jessica Lay Web Design. Well, look, her website got an image, got a favicon. Oh, look at that, beautiful, beautiful. We got a favicon there. Why can't we do that for the Hillstream Kingdom? I don't want the WordPress logo. It's a pet peeve of mine. Yeah, this. Come on. Come on. What was her name? Jessica. Let's get on it. Let's see, services, web design, SEO, logo design, graphic design services, and hosting services. Wow. Yeah. Website portfolio. Did the Hillstream people just not pay? What's going on here? Maybe they're difficult clients. Some of these other websites are looking. I mean, they look like websites. They look like they got favicons. Holy! All right, let me know. A couple fish that they redid the site. It used to be worse. Just a list of fish on a Google Doc. Elite Menagerie said the website's completely legit. Sam is a complete legend in the Hillstream community, and for once, I'd love you to do research before you judge people. Well, Louise, if you're listening, I've done no judgment. We're just researching live, and a couple of fish has already said these things in the chat. So this is what happens when you're just Googling and reading stuff. Holy. A couple of fish said I don't love any studios. I would say, let me go through here. The best, the one that's the most interesting is, now if it's the breeders is a pair of things, this becomes very interesting. Oh, did I say hi to Diana? What's up, Diana? A couple of fish said Sam is fine. The channel is good. His ID is bad. Balanced takes. I love it. I mean, whose idea is good with hill streams? It feels like it's kind of a very difficult thing that not many people are good at. I definitely would not be able to identify anything. Looks like a cool loach to me. Call it whatever you want. I'm gonna just have to go with it. All right. So, Mexicali, I have looked at the website for you. I've looked at the channel for you. It seems like this is a legit operation. We've got multiple cosigns in the chat, and the hill streams look cool. I say mission accomplished. Ooh, rice fish and half beaks. Ooh. Now hold on a second. Maros Harlequin half beak. Sexed pair. You have my attention. Fathom said, "Where's the blue banded Gucci sock loach?" You might have to make up that one, Fathom. I don't know. DP Exotic said in a couple discords, I've seen scientific papers that list Sam on them, so it should be pretty legit. Ooh, send some papers in the Discord. Let's check them out. Colorfish said he made some connections while studying there, if I recall. Wholesaler that most other hilly people already had also. Oh, Mick asked where he's getting the fish from. Elite Menagerie Habitat said, Private Mick, he has a partner in China. Sam is in the US, wholesales to retail and sells online, writes paper, and does symposiums on them. Deep Exotic says, I'm not going to lie, that half beak has me really rethinking how I want to set up a take, but I don't know enough about them yet. Well, I've had mixed results with half beaks. The celebs half beaks I had the most difficulty with, because it turns out, and I'm trying to remember because it was a little while back, but it turns out there's like a couple different Solese species that get mixed up. Some of them are more aggressive than others. Sometimes they come in like, I guess, mixed up, and they don't always play nice with each other. And I seemingly got one of the less friendly ones, and that was definitely challenging, because the platinum half beaks I've gotten some fry from them. I've raised those up. I'm trying to get my next generation going, but the Luis said, Nick bought his stock from me without knowing at the time that Sam was the source. Hey, well, those were definitely some fine loaches. I believe the ones that I saw earlier. Taintalumite Coboletum said, freshwater isopods win. We just went over the isopod paper a second ago, and we're just looking up random stuff right now, honestly. We're just looking up random stuff. I can picture, it was this one. This is the one that I got. So you guys have seen, I think it is. Well, Louise, you confirm. This looks like the one that I see behind me. I have to raise my camera. Ching Yun said, I love half-beak. I do love half-beaks. I'm trying to just focus on the platinum half-beaks. They look so good from above, I'll be honest. They're so shiny. A couple fish says, not private, my LFS is the same list, and several personal importers I know also buy from the same Chinese wholesaler. That's above my pay grade. Does that say 200? Yeah, it says 200, but it also says out of stock, so I don't know. Whenever something says a price and it says out of stock, it's kind of, I don't ever know if, you know, what the price was for. Okay. So, I'm gonna text Mexicali. Because Mexicali is the one that sent me this. He was like, he wanted to know if it was legit. Now, I will say, I understand why. I understand why, just because, you know, there's just small things with sites, like when a lot of stuff is out of stock, or if it's missing, you know, the little details, sometimes it's kind of hard. You know what a good example of this is? The current state of Aquabid. Like, the current state of Aquabid is like, let me get your credit card information stolen as quickly as possible. Like, look at this front page. Now, remember, when it initially relaunched, people didn't know they had to remake their accounts. So they're trying to log in. You see an ad like this, log into your account faster, easy login. You click on it. And there's actually some people I know that got by that. Not only that, but also entered their credit card information into things. Catgirl said always back to Aquavid. Yeah, but if you go to Aquavid now, it comes across as a much more... Like, the other site looked old, but it didn't look generic, because no sites look like this. This site looks like a lot of other sites, and it's kind of got ads everywhere. And it feels a lot more generic. But there's more stuff listed the last time I looked. It says 1,047 listings. Okay. I'm just looking at the killifish real quick, and then whatever you guys want me to talk about or look at, let me know. We can get into it. Whatever it is, I don't care. I just want to see it. The killifish, Aphysimian Kofi. Yo, these have been in a few auctions near me. I need to go and get them one day. Plefari, shiii. 3 wild pair, the wild pig, the Leptolucania amata. I had these in the 1 or 2 jump, but these are really cool native killifish. They're gold. They're on my list, but no new fish, Nicholas. No new fish. No new fish. 1 female blue galars. Look up some Ancestris SP. I don't know. Oh my gosh. It was 10/20/23 on the Kofi. Yeah, nothing too crazy. Nothing too crazy. All right. Oh, it is Friday. Did Wet Spot put out new fish? I just realized it was Friday. New fish. Let's see, let's see, let's see. Let's see, let's see, let's see. Nanochromis transvestitis. Let's go. Oh, you know what? I heard someone say it and it sounded more correct. It was transvestitus. And as soon as I heard that, I was like, you know what? That does sound right. That does sound right. Although I gotta give Wetspot credit. They never oversell the fish in the pictures. Because my goodness. That is a very undercolored Nanochromis transvestitus. Which is now how that's now how I'm going to say it. Very cool looking fish though. You want to build a native tank? There's some native fish that I do like. What suggestions you have? Let me hear. Let me hear. The native fish I have Elisoma gilberti. I have Lucania gudae. I have Heterandia formosa, or however you say. Do I have anything else? I think that's it. Yeah, I think that's all I have. T-bar cichlid, Nigerian red, pelvicochromis, Semophilus tremaculatus, black nasty, Cuban cichlid. I feel like, okay, wolf cichlid, angelfish. DP exotics. I thought about doing an orange-toed darter and northern plains killifish tank for natives. Cackerel said, I've been hearing a lot of big words tonight. I guarantee you, none of them were said correctly. Bitterlings is a great choice. Ooh, bitterlings are cool. They are cool. Have you kept them before? I don't know too many people that have kept them. They've been around in a couple of stores near me, but I haven't actually really seen them colored up in person yet. Silver cool. Oh, does anyone know about these guys? I was looking up these the other day. The Rhinochodorus woodsy. I don't know if I'm necessarily interested in, you know, one day having them or not. I just thought they looked really cool. Where's a good photo of these guys? It just kind of looked weird. I don't know, it caught my eye. DP Exotic says, I could legally collect both orange-throat darter and northern plains killifish in the same stream like an hour for me. You said Chinese perch for Munster fish? Chinese perch. Oh, God. You were joking about the Munster fish. Holy. That is a cool pattern, though. It's like a marbled fish. Wagyu perch. See, Aqua Imports has a red perch. Chinese red perch. Cineperca chauci. Adult size, 24 inches. That seems manageable. That seems manageable. Yeah. There's a slight 2 feet. I don't really see the problem. Nah, here we got some Facebook videos of them being really red. Damn. Nah. Facebook got the craziest videos sometimes of fish. Look at that. What the heck? It looks like the fish is ready for war. Look, the fish got the war paint. Minamano said the red video looks edited, TBH. Could be. It could be. Let's see. Predatory Fins has this video of them. The lighting isn't too good. They do look like they're pretty red in this. Honestly. I think they still look pretty red. Alright, I don't know what's going on with this. I don't know. Like, the saturation might be turned up, but I think they are. They do seem to be pretty red. If only you could find a video that's like... Ew. Oh, this is them albino. Oh my gosh. Oh, that is freaky. Ah! That is scary. Okay, I'm out of here. I'm out of here. That fish wants to eat me. Cool stuff, though. Cackerel said Kubatai Rasbora. Yeah, this is a good fish. It's a good fish. It's definitely a good fish. Fathom said they might not edit the video, but a lot of people use a red light in addition to their aquarium lights to accentuate the reds. Good point. That is a good point. See, Fathom's got all the tricks. Now you know if a fish look a little bit too red in Fathom's video, he's already told us how he's done it. He got the red lights. Come on. Cackerel said on the fish, simple, clean, perfection. That's how I feel about the hummingbird tetras. I like these guys a lot. They're like clear, but they got some. They do have color to them. Let me find a video. I'm getting a Depop commercial. Come on, bro. Yeah, I think they're nice. Deep Exotic said, look up Red Shiner, another local to me. I have thought about Red Shiner. Holy. Yeah, hummingbird tetras are they're great. I like them a lot. Mick said, love those hummingbird tetras. Ooh, I got something I want you guys to look at. 1 second. I need you to rate out of 42 the fish list of one of my local fish stores, okay? I want you to see what they got in. You tell me what you think. Because I was thinking about going there tomorrow, maybe, just to look. So I'm going to read the names and we'll look at some of the photos, but you tell me if this is worth visiting just to check out. So this is some of what they got in. So we got Chili Rasboras, which first of all, those have been gone for a while. We got Phoenix Rasbora, Giardini Arowana 8-inch, Royal Clown Knife 8-inch, Rainbow Shark 2.5-inch, Buna, Peacock, Black Lizard Loach, Dwarf Chain Loach, Black Chursidium Tetra, Corridorus Punctatus Napo Medium, Corridorus Shrilai Large, Loricaria Similimia, Tiger Knifefish, Pteroturosoma Microps, Hummingbird Tetra Small, Amazon Crystal Tetra / Pi Tetra, Red Atabo Pike 6 inches, Mesoharis Atromaculatus 2 to 3 inches, L168 Butterfly Pleco, L240 Galaxy / Vampire Pleco, L191 Broken Line Pleco 2 inches, True Parrot Cichlid, Assorted Discus, Vietnamese Lizard Loach, Reticulated Hillstream Loach, Golden Base Flowerhorn, Black Angels, Gold Lasers, Mouth Almighty, what the hell is that? Petricola, Brilliant Rummy-Nose Tetra, Cardinal Tetra, Harlequin Rasbora, Cobalt Blue Goby, Pygmy Corridorus, Skylar Mystax Barbatus, Panda Corridorus Small, Zebra Near Snail, Bolivian Ram, Holland Ram, Black Ram Balloon Body. Oh my god. Okay, well, let's see what that looks like. Azul Peacock Bass, Assorted Male Endlers, and Bamboo Shrimp. Let's look at some of these photos. Mick said red Ayabapo pikes are awesome. Ching Yun said, I found Kubatai could be a bit fin nipping. I used to keep them with Ember Tetras. Okay. First of all, I always think these knives look kind of cool. Okay, we got Discus. DP Exotic said lots of fish I've never seen in person, so I'd check it out. That's what I was thinking. I was kind of like, I have not seen some of these, so I wanted to check them out. 1 second. I gotta switch what tab I'm on because it wants me to be logged into Facebook. in order to view. Like, come on, man. I don't want to log into Facebook, man. I don't. Google Chrome. Nope, not that one. Give me 1 second. I gotta find the screen. Finding the screen that I wanna be on. It's taking me way too long trying to find the screen that I wanna be on. The screen share is taking too long. I'm clicking all the buttons when nothing is working. Oh no, I am certain I'm gonna make it. I gotta find it. Yeah, I got it. Let's go. I was getting worried. Lots of fish. He is a legend. Lots of fish. Is that a person? Alright. Here we go. Alright. Plecos. We got the plecos on the screen. Which ones were these? The peppermint? Peppermint? I don't know. Gotta go back to the list. Oh my god. The struggle of Facebook, man. Okay, the plecos listed. I don't even know if this one was on the list. Alright, let's continue. Arowana, normal rams. Oh, here we go. Mick is gonna love this. The black ram with balloon body. Oh my. This one's for the goldfish lovers. Wait, Mick said I want them all. You want them all, Mick? Are you sure? I got the balloon black ram on the screen. Come on. Maybe he's made an exception. Oh, Mick said perhaps not that one. DPXI said that thing looks so bloated. Well, I think that's the balloon part coming in. Got flower horns. Petricola. Now, which one is this? Which one is this? Does anyone know? Because I don't know. I feel like I've definitely seen this one before. One of them was called Mouth Almighty on the list, and I'm going to guess that this is Mouth Almighty. You're gonna see me live figure out how good I am guessing. Okay, I'm just gonna Google Mouth Almighty. Is this it? Let me wait. Did I actually guess it? Come on, man. Come on. It looks kind of similar. Yeah, I don't know, man. There's a lot of ugly fish out there. They might all look alike, but this photo at least is looking like Mouth Almighty to me. So I think I got it right. You guys are gonna have to tell me. I think I got it right though. Mouth Almighty is a crazy name. The flower horn is definitely a statement fish. Holy. Yeah, this is a pretty crazy look. I mean, flower horns, they do look nuts. I get the appeal. It's the hump that I can't get over. Mixed Glossamia aprion. Got whiptail. Is this some small? Is this the small peacock bass? I have to go back to the list. Okay, we got loach. These must be the royal lion plecos. Now this is the one that I want to see in person, but I'm worried that I'll go there and it's not going to be there because I think someone will have bought it. Because Wetspot's been running, or at least they were running ads on Reddit that featured this fish, and it had the text, "I bet you don't see this in your LFS," or something like that. And on the Reddit, someone posted on one of the aquarium Reddits like, "Is this fake? This looks like it's totally AI, because there's no way there's a fish that looks like a coolie loach," you know? But no, it's a real fish, so I do want to see. Okay, we got some cories. Yep, we got some loaches, cories. Man, chili rasbora, those might be gone. A lot of people keep it. I've heard a lot of people like in person talking about, man, I want chili rasboras, but they're all out of stock everywhere. Mick said I would feed those black rams to the glassamia. Come on, Mick. Don't be like that. I'm gonna see if you guys can see this. You're gonna have to really look close, but there's my. Oh God! No! The lights turned off. This is the tank that has my I guess my second, the Pistogramma nisonii that I spawned and raised that's now having fry is right here. I think you can kind of see. There's some distortion from the glass. And see, that's the other female I'm about to move. But she got it for first, what I assume is her first batch, a lot of fry. So one of the things I'm doing tonight is I'm moving all the fish out of there because she seems like a very active, aggressive mom. And, you know, don't want to stress all the other fish out. Don't want to stress her out. So I'm about to move everything. I was not planning on this occurring. It's crazy because the only male that was in there I just gave to Chad to complete his pair. But he left behind a family. But she looks like she's going to be a very good mom. Yeah, one of the things I've been working on is just getting all my tanks in order, sorting everything out, really getting caught up on any maintenance. And it's really been nice. It's been nice. So I'm making progress. I really want to get these orange crayfish going. I like them way more than I expected. Because it's kind of like, oh, small crayfish. I mean, I got shrimp for that appeal. But honestly, it's a little different. I mean, they're just bulkier. They got a different vibe, and the orange is really nice, as you can see. So color-wise, I mean, they're on par with all the Neocaridina for sure. And I just like them. I don't know what to say. I'm taken. I'm definitely taken by them. So the dwarf orange crayfish, so far, big fan. Real big fan. So a couple tanks I'm trying to get back into order. I'm doing decent sized water changes every day, gravel vacuum, just trying to get stuff situated. Trying to get my breeding setups, you know, solid. There's just only so much time in the day. So my schedule has been, I wake up at 6, I finish whatever packing I didn't do the night before, I go to work, I come back, sometimes I take a nap, water changes, pack orders for the next day, repeat. So I've just been tired. Mick says, August is crayfish season here in Sweden. Facts, no lie. So crayfish season, like crayfish eating season? Because come on. Minamoto said, I contemplate on getting those. Can they climb out the tank and escape? I was going to get the CPO and C Schultz Blue Morph. I spelled the name wrong. I mean, I just got them, so I'm not really any kind of expert on them beyond what I read. My understanding is they can, I think all the crayfish are going to be able to get out if you don't, or they have the potential to get out because they are able to crawl a little bit. And I mean, even shrimp can be prone to getting out of the tank somehow too, especially the bigger ones. So come on, lid game, get a lid. Mick said traditional crayfish party, one every weekend. Like, really? That sounds pretty fun. Not a crayfish party? What could be bad about a crayfish party? So, I guess I'll give a little update on this fish while it's here. Not that I have much to update, but this is what was sold to me as the Ornata Cotta, the licorice gourami. And it's going crazy right now. What's up with you? I'm sorry, I didn't mean to offend you. So here's kind of how it's been going with them. And I've been increasing, I'm trying to get the pH of this water down a lot. Cacro said, gonna stream, gonna hit the sack, I got homework to do tomorrow. They've been doing good, they've been doing solid. I've been feeding them live stuff, and they seem to eat some frozen stuff. The thing with them is, when I first got them, I bought 6. They were pretty skinny when I got them, and I did lose 1 or 2 initially. One had a swim bladder issue and just kind of couldn't really swim very well, but was around. After those initial losses, they've pretty much been solid, and they're the only things in the tank besides the Paradicinklus. I got 2 in there. So, as far as care for them, they haven't seemed like extremely sensitive or anything like that, just beyond the initial losses. But like I said, they were like very, very skinny when I first got them. I'm not really sure. I think I got 6. I think I have 3 now that I've had for a while. Not really 100% sure on the sexing of those 3. There was 2 that were doing the little gourami embrace, but it's just kind of hard for me to tell what's going on sometimes because the tank is full of utricularia. But that's what I got going on with them. I'm actually starting to like the utricularia as long as I can kind of tame it and just weigh it down with rocks. I just hate when it's floating and taking up the whole space, but like, I mean, it's a nice little mossy, algae-like thing for stuff to hide in, and it kind of looks cool with the crayfish on it. So I'm embracing it in some tanks, and I'm over it in others. I'm definitely over it in some places. Mix it all. That's making me sad to hear. Hope you got both. If they can't change, I mean, both genders that can't change. Yeah, I think I might. We're gonna find out. So this guy, you can see. Oh my God! As soon as I turn around, he moves. Sometimes you can see a little bit of red in his fin there. So I feel like he give me male vibes. And what makes me think the other one could be a female is I don't see the red and the same behavior. Kind of like being out and about, kind of territorial. Look at that guy's looking kind of great though. He's looking very, very proud of himself or herself. I think him though. So I'm feeling good about that. Let's see what else. What other failures? My failures have kind of slowed down. Successes are the dwarf pikes, the second generation of those going. That's pretty exciting. I've been wanting to redeem myself on those for a while. So I'm pretty hype about that. Actually, let me go check on, because the female had wigglers in her cave. Let me go let me see if they're still there. One sec. Yep. It was awesome. So the dwarf pikes, the Wallacea regani, I had a pair that had a bunch of babies a while ago. Those babies are raised up. Now they're having babies. So I just looked. The female seems to have at least 20 fry wiggling in the bottom of the tank right now. Johnny said, in my tanks, your baby isopods have shipped on hot weather, 80, 90s degrees. Would they make it? Probably. The only place so in all my time so far shipping isopods, I think I've only ever had 2 total losses of shipments. One, I have no idea what happened. Like, I don't know if the person was telling me the truth about what happened, but it wasn't even particularly hot, if I remember correctly. And she claimed, or he, I forget, the water was like 120 degrees or something insane. Like, it didn't seem possible to me. The other, again, was a little surprising, but it was in the desert, and I think the package got left out. That's only 2 times. I've shipped thousands and thousands of isopods, so not too bad. So you're in New York, I think. I would not be worried about it. I would not. When I do think it is too hot, though, I definitely do not send them. So there was a couple days, the last few weeks, that were just absolutely outrageous, and I was like, I'm not shipping any isopods in this at all. Mixed said, for licorice gouramis, females often lack patterns in the caudal fin and not as much patterns in the anal fin. All right. Let me go back to my crayfish watch real quick. I like watching the crayfish. Yo, is he about to attack the shrimp? Wait, is he about to attack the shrimp? Oh my god, is he sneaking up on the shrimp? No! That's a Malawa shrimp, crayfish. The shrimp's like, alright, buddy. You know you're bright and orange. I see you barreling towards me. The shrimp just slowly moved away. Okay. It looks like he was on some food that the crayfish wanted. That seems to be what just happened. We just watched someone get bullied off their spot. That's crazy. Johnny Best said, have you had any success on breeding crayfish? The big pink ones? Yes. The big pink ones, yes. These guys, not yet. Not yet. Not yet. This is I got a Luminatus pair. Well, I think it's 2 males and 1 female. So I'm going to pair them up for the upcoming auction. So I got those in there. This is supposed to be like a shrimp. Well, now it's a crayfish focused tank. But it's kind of like a crayfish shrimp assorted fry or holding tank. Temporary small fish hosting. When I'm going to move them somewhere. I am really happy about the Histatus though, because if you remember, I got a pair of, this started as a pair of Histatus. I think I did bap them, and now I have 10. Johnny Best said, aren't you afraid the crayfish might bully everything? No, not really. I'm not. I'm really not. If they get like a shrimp or something here or there, maybe that's fine. But I feed that tank quite a bit. My pink crayfish I've had with a few different fish, and it seems like it hasn't really been a problem so far. Those pink crayfish, they like eating plants. I'll be real. The pink crayfish, as long as there's plants in there, I don't think they'd ever bother anything else. I don't know how much these orange crayfish like plants in comparison, but they're small. The shrimp and stuff are pretty fast. That's all that's really in that tank, so I'm not too worried about it. Mick said that's a nice group of Hestatus. Yeah, so I'm enjoying the Hestatus. The group is kind of just growing. I want to move them to a bigger tank where it's just them and a bunch of moss. I think they make a bunch of Hestatus. They lay eggs on the glass. They don't seem to eat the eggs. They might eat the fry though. I haven't observed that, but it's definitely a strong possibility. I see 3 eggs on the glass right now. So, I'm pretty active. I've been feeding more frozen food recently. I like the frozen baby brine and like the frozen Moina, I think it was. Just because of how much one ice block has, and I can nicely pipette it, and it's small, and it just really floats around nicely. I'm gonna get my hatcher up. I need to buy some brine shrimp hatchery stuff, like from Mitchell, or from, I know Fathom's got some nice holders. I need to get the holders because I want to start hatching baby brine shrimp regularly again. Mick said you may need to move them if the cyclospina get aggressive. My plan is to move them pretty soon. So a lot of what I'm doing right now is setting up more species only tanks. So there's a lot of stuff being moved. Man, those are pistols having babies though. So annoying. I wasn't ready for that. I wasn't ready to move everything in there yet. There's not a lot in there. It's just the other pistos, and I wasn't ready to like be raising those fry yet. But we're gonna figure it out. We'll figure it out. But yeah, the Hestatus, I'm gonna move them because I want them to have a bigger tank to breed in. I want to try doing them in moths. Johnny Best, I said I was thinking the same, hatching brine shrimps. Yeah. Mick said, great to see you're doing the job needed. I need to do the same. I know. The one tank I've been doing a ton of water changes on the Nanochromis Borelius tank because the fry are getting bigger, and I had my internal filter in that tank fail because I was doing a water change and forgot that I was draining it, and it drained below the water level and it was running. So I'm trying to stabilize things in there. But there's a ton of Nanochromis babies in there. So the water change is probably helping them out a lot grow regardless. So I don't know, man. I'm doing my best. But look, I'm gonna get out of here. It was a good stream. Good little reading stuff. Good little brief learning more about some different places to buy hill streams. Shout out to Mr. Sam. Looks like you got some cool fish. Shout out to Wet Spot for the tiny small gourami. Something for us all to be fascinated with and read about. If only I wasn't too broke to buy them. Mick, shout out to Mick. Thanks for holding down the chat. Cat Girl, Big Steve, couple fish, appreciate you. Bob, appreciate you, Bob. Of course. Jeremy, Minimato, thank you for holding it down. Stopping by in the chat. Captain, Mod, and Chief, appreciate you going through. SBC, hope you enjoyed the genetics stream. DP Exotics, of course. Even Fathom came true. Shout out to Fathom. Appreciate you holding it down in the chat. In the Flesh Aquatics, how is it going? Hope you're doing all right. Oh, Diana did stop by. Appreciate you, Diana. Hope you've been doing well. Louise stopped by. Shout out to Louise coming in the chat, talking about wholesale stuff. Taintalumite, apologies you came in. We're done talking about the freshwater aquatic isopods. Xing Yun, shout out to you coming through, holding it down in the chat. Some native tank suggestions. The Chinese perch, it was a little bit scary, but we got through it. I appreciate you. Andy Carson, thank you for stopping by. Hope you're glad you enjoyed. And Johnny Best, which I can afford to buy them, is awesome. Although at the same time, even if you could, I would try and get the peppermint pikes for the same price. FUC said, great stream. Appreciate you, Johnny Best. Good night, y'all. I'm going to try and come back through tomorrow. Okay? I'm going to try. Stay happy. Stay relaxed. You know? Chill. Enjoy the weekend. Don't get into fights online, on the Discord, in the live stream, Facebook, Reddit, X. I said hi to Bob. Truth Social, Imager, Pinterest. What else? Where else can you get into fights? Some random form of Pistograma form. Hey, don't get into the all life fights. Relax for the weekend. Enjoy positivity. Eat something healthy. And I will see you when I see you. Alright. Something in my takes. And I'm out, right? I'm out. Mick said a pistol fights. That's a new one. You ain't been in no pistol fights. Come on, man. Come on. I've just been in my own little world recently. I'm so locked in. It's good. It's been bad for the stream, but I've been locked in, man. I'm in my own world. I'm in the zone, and I love it. I love it. Everything's just noise. She Savage Cichlid said, "Yo, what's up, everyone? What's up? How's it going?" One last selfless, well, not totally selfless, no, selfish. One last selfish plug. If you're in the DMV area, we're gonna be doing another auction, the Aquarius Society of Central Maryland. We're doing another auction August 16th. It's in Columbia, Maryland. For members, we have a 0% split on submissions, but we do take a $2 submission fee. So in theory, you should be able to make out pretty well. I think can I look at my last invoice to see my split from the last one? Anyways, regardless, I think for non-members it's 30% plus the $2, which I want to just do I just want to make it like 25% without the, I don't know. I gotta talk to them. I was, it's a group effort. The last auction went really well. We had some cool stuff. This auction is shaping up. I mean, it's a month out, so most people haven't listed anything yet, but this auction is shaping up to be pretty interesting. Aaron put in some Turkana jewels, some peacocks, some banded bars. We got some shrimp in here. Grace is coming through with some CW217s, the Hoplosoma rheatoma. 2 lots of 3 of those. Some rice fish. I'm bringing isopods and shrimp. I'm going to do some betta rubra. I'm going to see if I can get a pair of betta rubra together for this one. Some springtails. I'm going to bring a bunch of hill streams. I got a Riverless Pivot pair. I'm going to bring an Aphysemius striatum lamborine pair. I'll bring some Nanochromis Perilius. And I'm going to bring some Galaris from the Funge location. I got pairs of those. As long as disaster doesn't strike, I'll easily have a pair. We got, someone added some green dragon guppy culls. Talking about live bears a lot recently. Oh, sheesh. Parents of the Fry. Those do look pretty cool. That's Austin, right? Yep. Anyways, this is a month out. People are already listing stuff. So still plan accordingly. I think you should come through. I think you should bring something. She Savage Cichlid said, "Can you buy something? Send them to me and I'll pay you back." Oh, that's funny. Do they have German blue rams or black? I need some females too. I don't see any rams in there yet, but hey, come through, submit something. If you're a business or individual, you want to donate something to the club, send something through. We appreciate it. Last time, I think we covered the cost with a $2. I think how we did the math is like We don't need to take a percentage cut of sales. As long as we do $2 per lot and we get like 250 lots at least, then that pretty much covers the location. So then we have our raffle stuff and memberships and donations is what we rely on to make any beyond that. Is kind of the math. Because we're still going through the theory is we should be fine with doing stuff at cost or even slightly below and not prioritize the profit part of it. So far that's kind of working out. So we'll see. It's still early. It's still early, proof of concept, but last auction was a success. I'm having a good time. And our last meeting at Capital Exotic Fish was really fun. So I'm enjoying it. But just trying to stay on top of it. I don't think I'm the best aquarium club president ever, but I do think we are doing a good job of helping each other in our weak points so far. But I do think we'll probably need to get some more people on the board if we want to keep maintaining and doing a good job. I'm watching my panda Corey just laid an egg in front of me. But yeah, anyways, Aquarium Society of Central Maryland, August 16th, Columbia, Maryland, come through. There you go. I'm headed out in my tanks.